So transcription is, of course, the process in which DNA is copied to RNA. And the RNA is subsequently translated into proteins. And in eukaryotes, again, the packaging of DNA into nucleosomes and chromatin needs to be handled. And, certain pieces of the DNA are very rarely transcribed. Some parts probably not transcribed at all, while others are very frequently transcribed. So how is the nucleosomes arranged according to the need of frequently transcribing these genes? The eukaryotes are forming the beads on the string, the nucleosomes on the DNA, and the DNA needs to be exposed. Now, if you have a start site for transcription here, then it turns out that the nucleosomes are bound in a very, say, statistically significant manner. So there is a wider gap at the transcription start site. There are usually no nucleosomes bound to this area. But after the transcription start site, the nucleosomes are very, with high frequency, positioned at a certain distance here, and then while you go further and further away, it becomes less, more and more fuzzy. And on the other side, the -1 nucleosome is fairly well defined, but the positions beyond that is again more fuzzy. So the transcription, of course, goes on in this direction, and this is the transcription start site. So the open area here is in the order of 200 base pairs. Okay. And to then operate and make the genes available for transcription, we have the nucleosome modifiers. They act to make certain genes more accessible for transcription. And there are several ways to change the position of nucleosomes. Nucleosomes can be sliding. So this nucleosome is moved. So it becomes a more open structure here. You can also remove nucleosomes, so you get a gap here. You can also assemble nucleosomes onto the DNA and add one to become this type of structure. And the spacing of the nucleosomes can be rearranged this way. And there's also different enzymes involved in replacing histones, specific histones. So say the H3, H4 remain here, but some yellow histones are added onto this, and some of the blue ones are removed. So there are different histone variants that can be replacing the normal ones. So there's a whole big field of how to handle the nucleosomes on the DNA here. This picture shows essentially the same thing. So here is how you deposit a new nucleosome onto the DNA, and there is remodeling proteins involved in this using ATP. So you're pushing these nucleosomes a little bit apart to add another one. Here there's a double DNA binding protein which binds to this and which could be then used in different ways to, say, make new arrangements of the DNA. So this is basically showing the same thing as the previous picture, but with a slightly different outline. And transcription in eukaryotes is complicated by the phenomenon called epigenetics. And the epigenetics is, first of all, involving the modification of DNA, the methylation of Cs. but also the histones are modified, and this is very interesting because the histones are primarily methylated, but they're also acetylated. And actually, there are a number of other modifications of histones, but we cannot, in a short lecture, go through them all. But these modifications make the genes accessible or inaccessible for proteins engaged in the expression of genes. So there are then readers, proteins which are reading the histone message to see, is this something which should be transcribed, or is it something we should skip? And these modifications on the histones are to a certain extent inheritable. So the histone code determines which genes will be transcribed to some extent. Now, here's then histone H3 and H4. And you remember from last hour that they have long amino terminal tails. So here is the tail of H3, and it's primarily the lysines which are of interest here. Lysines and arginines, as a matter of fact. So here is lysine number 4 in histone 3. And so if you remember, I said these amino acid sequences in the histones are very, very highly conserved from one species to the next. So lysine 4 is probably lysine in most species. And this lysine can be monomethylated, can be dimethylated, and trimethylated. And so these modifications are very important because this is what the readers will read. Again, lysine number 9 can be modified. Lysine number 27 can be modified. Lysine number 36. And in the histone 4, the lysine number 20 can be modified. So making a certain pattern of this will, say, be a signal which can be read by the readers. So the N-terminal tails here of the histones, they're involved in the interactions with the DNA. They interact with other nucleosomes. And this is H3, and here both H3 and H4. And the lysine modifications, methylation, acetylation, phosphorylation, ubiquitination, summoylation, control the transcription of the genes. So these are a little bit bigger number of the modifications you can have. So some of the modifications here. H2B has 12 different modifications, and it's lysine 5, lysine 12, lysine 20, and lysine 120 that can be acetylated. H3 has 21 different modification possibilities. Lysine 4 can be acetylated. Lysine 4 can be methylated once, twice, or 3 times. And lysine 9 can be acetylated or methylated once with one methyl group. Lysine 18, lysine 27, and lysine 36. And H4 can have 14 different modifications. So there are specific enzymes modifying specific lysines in the histones to control the transcription of the genes bound to them. So these are the writers. So there are specific writer proteins that make the modification. Either they can add 1, 2, or 3 methyl groups to the lysine, or they can add acetyl group to the lysine. And this, as I will show you, become quite different. And as I said, or I didn't say this, likewise there are specific enzymes removing the modifications. These are eraser proteins, removing the modification. So acetylation is normally associated with transcription. Acetylation makes it easier for to remove the histones and to access the DNA. But methylation is often associated with silencing. But it's not, say, completely black or white. It's there is a bit of a bit more complicated than just acetylation is associated with transcription. Sometimes it may also inhibit the transcription, but generally acetylation goes with transcription, methylation goes with silencing. So readers. The methylation of lysine retains the +1 charge, because you remember the lysine side chain has a 1+ charge. The amino group is positively charged. And if you methylate the lysine with 1, 2, or 3 methyl groups, it remains positively charged. So in the interaction between proteins and DNA, the charges are, of course, very important, because you cannot, say, put a charge into a hydrophobic environment because it needs to be stabilized. It will be a very unstable situation putting a charge into a hydrophobic environment. You need to have something that balances the charge. So 3-methylated lysine is positively charged but hydrophobic. So this is very strange. It has no possibility to make a hydrogen bond, but nevertheless it is having a positive charge here. I mean, imagine the amino group having 3 methyl groups on it. It is positively charged but cannot make a hydrogen bond. So how do you deal with this? And I'll show you that. So different reader domains interact with differently modified residues. So lysine methyl 3 group binds into an aromatic pocket. So this is the way to deal with it. And I showed you yesterday how the negative charges on an aromatic group are floating above or below the ring structure, having partial negative charges, and there's positive charges around the edge of the ring. So this is the elegant way to deal with this. So here is then a 3-methylated lysine in a specific reader domain, and look how it's organized. So this is the peptide on which the lysine is sitting, but here are 3 aromatic groups surrounding the lysine, and they're all showing the negative charges towards the lysine, partial negative charges, so it balances the lysine positive charge. And here we see the arrangement of the polypeptide, same polypeptide. So it makes a beta structure. The polypeptide is recognized by this chromo reader protein. The chromo is the reader protein here. It makes a nice beta arrangement, but the lysine is then finding a proper pocket where it can stabilize its positively charged that is 3-methylated through the aromatic arrangement around here. This is another situation where we have a lysine on histone 4, lysine 20, which is doubly methylated. So this has then 1 hydrogen left open. So only 2 methyl groups on the 3 positions. So it has the possibility to make a hydrogen bond. And actually there's an aspartate here, which makes this hydrogen binds, becomes the acceptor for this hydrogen bond from the lysine. But otherwise, there are here 4 aromatic groups, which again provide some of the negative charge to this positive lysine. Methylated lysines have fewer hydrogen bonds to satisfy, and 3-methylated lysines, doubly methylated lysines, and singly methylated lysines, and not methylated lysines. They bind to different sites. So different enzymes recognize, well, this is a doubly methylated, this is not methylated at all, this is 3-methylated. So, I mean, one single lysine here can give a number of different messages, and it will interact with different reader proteins. So the histones then become a very elegant way to give messages to the system. In addition, there are methylated arginines. They can have one methyl 1 or 2 methyl groups on the arginine. In the latter case, they can be symmetric or asymmetric. And this RME2S is symmetric, and RME2A is asymmetric. And there are specific reader proteins, again, that can differentiate between these different situations. But the reader domains here, they have a limited set of folds. There are many, many, many reader proteins, but the number of folds, they are, say, relatives to each other. They're cousins to each other. So the fold is the same, but the specific arrangement around the site which is recognized, the modified residue, will be different. So some of them are called chromo, others are Tudor, others are PhD. So there's a long list, but I picked the most frequently occurring ones. And sometimes they occur as single domains, but often they are, say, several domains in a protein which can read a number of different modifications. So they're looking for some specific set of modifications, knowing this is my site, because all the domains will find the proper reading message. Different reader domains to affect the transcription of specific genes. The acetylation of lysines make the residue uncharged, but more hydrophobic. So acetylation is most often associated, as I said before, with transcription, and deacetylation is leading to repression. So here again we have a modified lysine, which is acetylated, and here you see the amino group, and here is the acetyl group, and it's now not charged, and it's binding to an asparagine here, which recognizes this very long arrangement. So nothing else would be able to make this binding. And here we see this is the same case shown, I think, in 3 different orientations. So here again you see the hydrogen bond, but you see also the whole peptide, how that is arranged, again with some type of beta arrangement. It's stretched out like a beta strand. And also here there is an arrangement of aromatic groups around the hydrophobic lysine. So here we have 2 connected domains which read different messages. So this domain, the PhD domain, reads histone 3, lysine 4, 3 methylated, and this domain over here, the bromo domain, is reading the histone 4, lysine 18 acetyl group. So, I mean, the combination here of these domains makes it very specific reading. Yes, this is my perfect site where I should be. Then we come to a different field here, and this is the gene-specific factors, because, I mean, maybe I have not emphasized it enough, but you don't want to express all the genes all the time. The genes are expressed when there's a specific need of them. So if you take, say, a newborn baby, it has very specific needs for genes, and an old man like me, I have entirely different needs of expression. So you need to have very specific means to activate the genes that are needed. gene-specific transcription factors. And there are also both repressors or activators that control the transcription of the different genes. And there are a number of, say, repeated motifs that we see in these type of things. So there is some different, the names are given here and the description of the domain is here. So B-zip is basic lysine zipper motif. BHLH, basic helix loop helix structure. And this can be combined. Winged helix is another different variant. And then zinc fingers, there are different type of zinc fingers. There is C2H2, which means cysteine, 2 cysteines and 2 histidines bind a zinc. And there are zinc fingers with 4 cysteines. There is also 2 zincs bound to 6 cysteines. And then the homido domain, and there's a stat domain. So we'll show some of these examples. So here is then a beautiful arrangement of the coiled structure of piece of DNA binding to the major grooves of the DNA here in this specific way. And if you look into the details here, the helices here, they recognize have hydrogen bond connections to the protein. So this type of protein is called a lysine zipper because the residues that interact along the helix here is quite often lysines. And actually, if you remember now from yesterday, there are 3.6 amino acids per turn of the helix. So if you go 3 residues down, you don't really say, if you start from this point here, that is facing the other helix. But if you go 3 amino acids down, you will end up somewhere here. And if you go 4 residues down, you'll be somewhere on the back side. But if you go 7 residues, then you're again meeting the other helix. So what has been observed here, that this type of coiled structures can have lysines, hydrophobic lysines, every 7 residue. And then the lysines will be very happy to say, form like a glue between the helices. So a lysine here, lysine there, and then 2 turns down on the helix, and yet another pair of lysines over here, and so on. So there can be 3, 4, 5 lysines being 7 residues apart down the sequence. And as I say, they form some kind of glue holding the helices together. So here is some other type of lysine zippers. This protein is called MyoD. It has a little bit different type of structure. So there's a helix loop helix arrangement here, and also here the MYC and MAX proteins. They look in this way. They interact with helices in the major grooves of the DNA, but they have slightly different ways of dimerizing. So these are all dimers because the DNA is basically, in this case, you can see that the DNA is slightly bent also. So this crow and the repressor here, they come from bacteriophage lambda. It has 2 repressors that bind to the operator region. And the second helix binds in the major groove. Second helix of the protein binds in the major groove. So both these are 2 different repressors in the bacteriophage lambda. And here I think we have the trip repressor because, say for instance, now of course a cell needs to have produce tryptophan. So the genes are involved in producing tryptophan, they should be operative. But if you now have a high concentration of tryptophan here, then you need to shut off these genes because why should you transcribe them again? So then this is the structure of the triple repressor without tryptophan bound. And in this case, it doesn't repress. It just sits around. It has the wrong conformation to bind to the gene. But now, if you have a high concentration of tryptophan, the tryptophan binds here to the protein. It changes the structure just very slightly. If you look at it, these red helices, they are a little bit inclined in this way. But when tryptophan binds, they open up. Now it can sit down on the DNA. So the blue helix gets into the major groove again. So this is, say, a simple way that many genes are operating, or many of the repressors are operating. If there is a high concentration of the compound that is produced by the genes here, then that will inhibit in this way, repress the transcription of the gene. So here are some more turn helix motifs. The MUT1, MUT2. So this is a heterodimer binding to the DNA, and here you have OCT1, which is also helix turn helix motif binding to the major groove, and PU1 helix in the major groove, just to show you a number of examples. Now we come to the zinc fingers here, and here you see a zinc finger protein binding to the DNA. Actually, this protein has actually 3 zinc fingers in a row, and they all bind to the DNA, finding a very specific site. Here you see that this is a zinc finger with 2 histidines and 2 cysteines bind to the zinc. And the glucocorticoid receptor is a zinc finger. Again, you see here, this is a dimeric protein, and you see 2 zincs in each of them. And this is a zinc finger with 4 cysteines binding to each of the zinc ions. And as I've been pointing out, the specificity of interaction between the protein and the DNA is coming very much through hydrogen bonds. And here you see some specific hydrogen bonds which identify a specific sequence of the DNA here, because, well, I don't think I brought a figure of this, but the DNA sequences, they make hydrogen bond donors and acceptors in a very distinct pattern. So 3 or 4 nucleotides in a row here would make the pattern very, very specific, and a protein can recognize then a specific sequence by having the opposite type of acceptor or a donor for hydrogen bonds. So this is one case, this is another case. You see lots of hydrogen bonds here between the protein and the DNA. So, I mean, proteins can read, to some extent, the DNA sequence. Now, we come to the real thing here, because we have the DNA here, and somehow this will be transcribed into RNA. And with the help of, first of all, removing the nucleosomes or opening up the sequence, and also recognizing some specific sequence here to which the RNA polymerase should bind, then we know that now we are about to start to transcribe the correct material. And the genes are organized a little bit uniquely. So here in bacteria, we know that we have a gene here, and there is different boxes preceding the gene to be recognized, so that we do the proper job and start in the proper place. So there is a box here, the Privnov box, with a sequence which is most usually TATAAT. And this is about 10 base pairs preceding the gene. And then there's 35 base pair away you have another sequence, TTGACA, which is also very important signal for recognition of the correct promoter region for the specific gene. For eukaryotes, it is, of course, more complicated. So the gene itself here is composed both of exons, which are going to be translated finally, but there are also introns, a number of introns, which are not going to be translated and which are removed by the spliceosome. And I don't have the time to go into the way the splicing is done, but now we are getting quite far into the mechanism of splicing also. And but in eukaryotes, there are regulatory elements both beyond the gene and far before the gene. And in the promoter region here, which can be 300 base pairs long, there's a specific box called the Tata box, which is about 25 base pair before the gene starts. So now we'll look into how this is organized and handled. Well, if you want to transcribe something, then you need to open the double-stranded DNA up. So you make a transcription bubble somehow. So this is the upstream DNA. Here's the transcription bubble, and then the downstream DNA, which is not yet transcribed. But here's transcription going on from the active site, which is here, and then there's a piece of messenger RNA being produced here. So you go in the polymerization direction to the right here, but if something has gone wrong and identified somehow, the enzyme can also backtrack. And in such a case, there is different pieces of the RNA, or longer or shorter pieces of the RNA coming out through a specific channel. I'll show you how that looks, which will be degraded such that you can make a new start, make use of the proper piece of the RNA, but degrade the one which is faulty in some way. So first, a little bit about the bacterial enzyme. So these large enzymes have been investigated by crystallography, and the bacterial enzyme is composed of 5 different subunits, 2 alpha subunits and 2 beta subunits, beta and beta prime, and the omega subunit, and you have them in different colors here. So alpha and alpha over here, the beta is in yellow, and the beta prime in green, and the omega in blue. And here is then a factor, transcription factor, sigma, which is sitting over here. And this has also been investigated with the bound DNA. And you see here the leading strand in blue, the lagging strand in green, and the red is the RNA produced. And this complicated protein here, called sigma, because it's complicated because it doesn't make a simple domain held together, but there are various pieces. You can say there are 3 domains, blue, green, and red, and there is the stringy part over here, which are all needed to activate the transcription. In eukaryotic world, there are 3 RNA polymerases, pol 1, pol 2, and pol 3. Pol 2 is the one that's dealing with producing the messenger RNA. But the pol 1 is making ribosomal RNA, transcribing ribosomal RNA, and pol 3 is transcribing tRNAs. So all 3, of course, are needed to make the protein finally, because we need both ribosomal RNA and tRNA and messenger RNA to make the protein. And it's of course interesting to know that these enzymes are very strongly related to each other. Bacterial RNA polymerase and the 3 eukaryotic versions of RNA polymerase are all having similar subunits. They are not identical, but they are similar. So here are the different names for the different ones, and the archaeal RNA polymerase is also very similar. So there are some conserved subunits, say the bacterial subunits found in bacteria, they're conserved in all other organisms, but then there are some non-conserved subunits. And this is then the eukaryotic Pol2, RNase polymerase 2, which has 12 or even 14 subunits. And here you see the leading strand of the DNA and the lagging strand in green, and the red is the RNA produced. So here is all 3 enzymes have been investigated, Pol1, Pol2, and Pol3, and in general terms they look quite similar, but in details they are a bit different, and we don't have the time to go into all of this. So RNA polymerase is able to synthesize RNA, but it is unable to identify a promoter and to initiate the process. So this needs a great number of helpers, general transcription factors, general GTFs, and they are given the names TF2A until H, B, C, D, and so on. And there are about 60 proteins which are needed to perform RNA synthesis. So the first task is to identify a promoter and bind the DNA to RNA polymerase, and to generate a transcription bubble and to place the transcription start site TSS in the active site and then start RNA synthesis. So there are a number of tasks to be solved, and of course this has been done billions of years ago. So the general transcription factors here are TF2A, B, D, and here is a very important protein, TF2D, or actually TBP, Tata box binding protein, very important protein. A small protein, 30 kilodaltons, binds to the Tata box of the promoter. And this TF2B is also a very important protein, as one subunit, and it's not so big, 40 kilodaltons, and it's central in the pre-initiation complex formation. Selects the TSS, the transcription start site, and brings the Tata box DNA near the active site of the RNA polymerase. Then we have the so-called tufts, and there are 13 of the tufts, and the total mass of them is 880 kilodaltons, and it assists Tata box binding protein in recognition of promoters. TF2E, 2 subunits, TF2F, 3 subunits, and this is the helicase involved in strand separation. No, sorry, it's helping in strand separation, but it's TF2H, which is the helicase, and actually this 1 subunit out of the 11, SS12, which is the actual helicase. And then we have something very big here, mediator, which has 25 subunits and a total mass more than 1 million, which interacts with transcription factors, with the gene-specific transcription factors to mediate the information to the RNA polymerase. So it's quite a complex system. So now let's look at the Tata box binding protein. And it is a funny and interesting structure. It looks like a saddle riding on a horse, or laid on the back of the horse. The horse being the DNA. So here the DNA is then bent by the binding of the Tata box binding protein. And there are certain elements here, we see them here, that are on each side 2 phenylalanines of the Tata box binding protein, the dimeric protein, the green and yellow subunits. These two phenylalanines are inserted into the DNA or intercalated and making the bend of the DNA. And the saddle is sitting and across here bending the DNA. And this bend is quite important for the future function. So now this is a very sketchy picture. So the Tata box binding protein is sitting here and bending the DNA in this way. But this is not the functional arrangement yet of the whole thing. You see the RNA polymerase in gray here. The active site is down here where the RNA should be produced. So this is not the way to bind it. But the TF2B, this reddish structure here, is the factor which is involved in rearranging the DNA such that it opens up the bubble here, and you have placing the leading strand into the active site, and the lagging strand is not seen in this type of structures until we come a bit beyond the active site, and here we see the non-template strand. Okay, so the Tata box binding protein and transcription factor 2B are two very essential ingredients in forming the correct structure here. So TF2B identifies the transcription start site, it forms the pre-initiation complex, it interacts with Tata box binding protein, and actually it corresponds to the sigma factor in bacteria. So to make a simple comparison here, here we see the TF2B in its place on the RNA polymerase. This is just part of the polymerase called the wall structure, and here is another part of the RNA polymerase. You see both in the bacterial case and the eukaryote cases, you have these RNA polymerase structures being quite identical, but the proteins here have very different arrangement, but they do the same type of job. So they put the DNA, the leading strand of the DNA, or the template strand of the DNA in the proper place by different type of structures, but they are, say, correspond to each other functionally. So here we see the TF2B and we see the strand of DNA here with a sharp kink produced in the active site. But as it says here, once we have made roughly 8 nucleotides of RNA, then this collides with the TF2B, and TF2B will have to dissociate and fall off from the RNA polymerase, because now it's done its job and everything is in the proper shape to be proceeding. So this is then repeating what I've said already. So in some cases, the Tata box, not all promoters contain the Tata box, and but if you have the Tata box, they only need the Tata box binding protein of 2F2D. But if you have Tata-less promoters, they also need the different tufts of 2F2D. I mean, the 13 different subunits, which are helper subunits. The opening of the transcription bubble occurs about 20 base pairs downstream from the Tata box binding region, the Tata region. And the rigidity of the DNA requires the melting of the DNA before it can adopt the 90-degree turn that we have down in the active site here. So the DNA has to be melted, and this is partly due to TF2B. So here we have the different steps in incorporating a new nucleotide. So here we have the downstream, and here we have the upstream DNA. Here's the active site, and here's the base that now will be transcribed. The red stuff here is, again, of course, the RNA. And there are various parts of the enzyme which is very important. The bridge helix in here and the trigger loop are in the active site and being very important. So here's the nucleotide coming in, an RNA nucleotide, 3 phosphates and the base and the magnesium ion. So they come into place here and are being, say, trying to base pair properly with the base in the active site of the DNA. And if they're correctly identified, the Watson-Crick base pairing, then the whole thing proceeds. Now it's let into the active site properly, and then they are connected this way such that also the two phosphates are released and they disappear together with the magnesium. And during this process, there are some conformational changes of the active site, so this trigger loop becomes 2 trigger helices. So nucleotide by nucleotide, you go around in this circle, add one after the other. So to look at it in a little bit more detail, so here is then the incoming nucleotide, which is base pairing with above with the DNA, and so here's the triphosphate, magnesium, and then they're moving close to each other, and then this phosphate is getting associated with the ribose here, so you get incorporation of new nucleotide. I mentioned the case where the RNA is being backtracked. You move backwards because there is some problem of the RNA produced, and then it comes out through this funnel on the lower side, and then there's a specific transcription factor 2S, which is an RNA nuclease, so cleaving off the nucleotides which are coming out through the funnel below, which is the same funnel through which the nucleotides are entering the active site. So a summary of the process of transcription. Specific transcription factors bind to a gene, and they interact with acetyltransferases to make the gene more accessible. Histones become acetylated, less tightly bound, and the mediator, the 25 subunit mediator, interacts with the transcription factors. Histones are removed. Histone chaperones can put the right ones back again. The gene becomes accessible, and the chromatin remodeling proteins using ATP have, of course, assisted in this process. The Tata box binding protein of TF2D identifies the promoter and binds to the Tata box and bends the DNA, as I have shown you. And then the tufts of TF2D may have to interact with the bent DNA of the Tata box, and TF2D is stabilized further by TF2A. Now it's time for TF2B to come into the picture, which interacts with the Tata box binding protein and TF2D and positions the promoter on the polymerase. And this forms then the pre-initiation complex, PIC, with about 32 different proteins being involved. Then the 2 DNA strands have to be separated, and the proteins involved in this is 2F2E, TF2F, and TF2H. And the helicase TF2H has a central task in this helicase activity, and ATP is consumed here. Then you need to place the template DNA strand in the active center and identify the transcription start site. And TF2B is the important ingredients here. And then RNA synthesis can begin. So then if you have the promoter here, and you then have a whole complex of proteins which bind here to identify the Tata box binding protein and so on, and then the RNA polymerase can travel down the gene. And of course, you can have several RNA polymerases moving at the same time. They are initiating here, but then they're traveling along the gene, and several RNA polymerases can be active at the same time on the same gene. And I think this was the very end of this lecture. So now